Excuse me, ma'am. Wala po'y audio.
Okay, thank you for that. Yo-yo na ko na. So the bottom line again, when we are talking of the Oh, okay. So the bottom line again, when we are talking of the alpha helix structure is on the identities, no, on of the amino acid, because the R groups also affects on the stability of the alpha helix. So whatever the identity of the amino acid sequence and the identity of the amino acid and the sequence also will determine on whether the polypeptide sequence will assume an alpha helix or not. Okay, and these are some of the considerations, no, that will affect on the stability. If there is a repulsion, of course, to stabilize. So whatever that could stabilize on the alpha helix would lead to an alpha helix structure. And again, another is the R groups, no, it will affect. So whatever that could stabilize the alpha helix could lead to the alpha helix secondary structure. And the bottom line is on the identity and the sequence of the amino acids. Because if we alter the sequence of the amino acids, and for example, two R groups that are close proximity within 4 residues away could destabilize. So therefore the alpha helix will not be formed anymore. So the alpha helix, just an additional note, that the alpha helix is an additional dipole. It's an alpha helix, a dipole, rather. There is a dipole. Ano sa mga dipole? Dipole is no, when you have a net charge. You remember your general chemistry, when you have a net charge. So negatively charged amino acids often found here to stabilize the positive charge of the dipole. So here you have amino terminus and it's passively charged. So any close proximity that are negatively charged amino acids could stabilize that one. And on the carboxy terminal end, you have negatively charged carboxyl terminus. So positively charged amino acids coming side chains. There's no side chains here. Close to that could actually stabilize the negative charge dipole here. And then you have, of course, I have already mentioned that there is intramolecular hydrogen bonding. You have hydrogen bonding every 4 bonds. Let's see, hydrogen bonding here, no? And that hydrogen bonding actually is arise from the dipole, no? Ang ng dipole, na naka-net charge due to the electronegativity of the some atoms, no? Katong mga atoms again they have mentioned, katong if you have oxygen, nitrogen, no? Those are electronegative atoms and they will actually assume a dipole, and depending on its orientation, if whether they have a dipole or not. So again, this will be affected, or this will depend on the sequence of the amino acid I have mentioned and the identity of the amino acid. Another secondary structure of the amino acid is what we call as a beta conformation. The beta conformation of the beta sheets is formed again through hydrogen bond, no? Parehas sa alpha helix hydrogen bond, but if you look at its structure, murag in ani, naka-sheet siya, no? Wala kay helix makitaan. So the beta sheets is formed in 2 or more polypeptides chain segments lined up side by side. So if you have an amino acid, no, like left to right, amino N terminus and C terminus N or carboxyl terminus N. So nakay polypeptide sequence, and then na siya silingan nga polypeptide sequence gihapon from N terminus to the C terminus N. Okay, you have that. And then they are in close proximity, no, to each other. And there is what we call as an interaction between this amino acid sequence or polypeptide chain lined up side by side. In the beta conformation, the backbone of the polypeptide chain is extended into a zigzag rather than a helical structure. So you have like a zigzag structure here, side chain, nag-line up ang chains of amino acid or chains of polypeptides. So nag-line up sila side by side, and they are stabilized by a particular weak forces of interaction. And that forces of interaction is actually just also a hydrogen bond. So each individual segment kaning sheets is referred kani siya is called as a beta strand. So beta sheets are stabilized, as I have mentioned, by hydrogen bond. So asa man ang hydrogen bond? Earlier we've learned for the alpha helix that the hydrogen bond is intra. Now, when you say intra, it is within the amino acid sequence residue, kanang within the chain lengths of the polypeptide sequence, no, naa dito ang hydrogen bond. Every 4 turns mo undergo hydrogen bond ang kanang 4 amino acids away, no, it will undergo hydrogen bond. But in the that will stabilize the alpha helix. In the case of a beta sheets, nagyapoy hydrogen bond, but the hydrogen bond is formed between the polypeptide backbone. So let's say you have the arrow is written from down to top, so it's okay if you just indicate on the arrow. No worries the N. So you have the N terminus to the C terminus, okay? For example, here you have the C double bond O, you have an oxygen here of your peptide, C double bond ONH, and the hydrogen of another amino acid sequence or amino acid polypeptide, or rather a polypeptide chain here. And there you have a hydrogen connected to the nitrogen. So you have to remember that for a hydrogen bond to occur, the hydrogen should be connected to an electronegative atom such as nitrogen, oxygen, and fluorine. So you have a hydrogen connected to nitrogen, an electronegative atom. And this is very partially positive, and you have an oxygen and electronegative atom. So hydrogen and electronegative atom, this actually forms a hydrogen bond. So there is a hydrogen bond formation sideways, no, from between the, again, polypeptide sequence. So very unlike the alpha helix that it is intra, which means intramolecular hydrogen bonding just within the molecules or the sequence of the polypeptide. This one is intermolecular. This is in between chains of polypeptides, chains of amino acid residues. You have a hydrogen bond here. You can have hydrogen bond. You can also have a hydrogen bond. So you can, between this, there are two sequence of amino acids. It will actually give rise to a beta plated sheets, stabilized again by the polypeptide backbone. You have here polypeptide backbone or your amino acid sequence residue. And just have to take note that the beta conformation or sheets actually are made up of two kinds. Now they are antiparallel or parallel. Okay, what is the antiparallel or parallel? Wala gibutang, but here I will just say, no, if it's parallel, no, the sequence of the amino acid polypeptide backbone is similar. Let's say start from the C terminus to the N terminus, parehas siya og direction, C terminus, and the other polypeptide backbone also has the sequence C terminus to N terminus. So if you encounter that kind of polypeptide backbone, adjacent to another polypeptide backbone to form a beta sheets, then it is in a parallel beta sheets form. But when the C-terminus of one polypeptide backbone and the other is in opposite direction, no? Like here on the left, you have the N-terminus down to top. No, your N-terminus is na diri, pataas. And the other one is the N-terminus is nasa taas pa down. Then this is what you call as an antiparallel beta sheets formation. Okay. So here is the top view. This one is a side view of what the beta sheets is. It's like a zigzag structure. But you look at on top view, you have this. No, it's kakuan mo anang murag corrugated no cartoon. Murag inana. So this is what you call again as a beta conformation. So it's like a zigzag structure and it's like a corrugated one view on top. Mura siyang seam, no? If you look at the seam in ani siya, sheets. Kanang corrugated na seam. Okay? Now, sometimes in the secondary structure, dili lang kay beta or alpha. Sometimes both the alpha helix and the beta sheets are combined. So, for example, here is a class of protein where you can find our binding sites here. This is actually this is the alpha helix. The alpha helix, kaning murag ribbon. Okay, this is the 3D model, 3D structure of your amino acid polypeptide sequence. No, so here you will notice here that you have the plain murag ribbon na plain. This is actually our beta sheets. No, and you will notice that isa lang siya ka sequence rather. Yeah, so it still can be, it can also be intra, no? Pag isa lang ka sequence of amino acid. Notice na continuous ng sequence of amino acids, but very few amino acids away, pag magsilingan sila, it's possible to have the beta sheet. So you have a plain here, ribbon, straight ribbon up. So you can have here an antiparallel beta sheets. Now the amino acid sequence is pana, top to bottom. This one is bottom to top. No, so there is a beta sheets here, which is antiparallel. This one is not part of the beta sheets. So we'll discuss later. Those are just coils, no, that has no organized structure that would lead to beta sequence or beta sheets or alpha helix conformation. So this one is also amino acid chain or sequence, no, but the chains of amino acids here does not lead to a particular alpha helix or beta. So this is an example of your protein containing both your alpha helix and your beta sheets. So note in this example how the alpha helix can gradually curve and are not exactly straight. You can see here, no, it's not that straight as you have in the example. Again, it will depend on what are the R groups are that made up, what are the R groups of the amino acids or the amino acids identity itself, no, in this sequence could affect whether they could form like a perfect helix, like straight ribbon, or a some kind of turnings gamay, no, like for example, you can see little bit dako nga turn and the other one is shorter, but it's always around 4 residues away that the hydrogen bond is formed to stabilize this one. Okay, and then here they lost now already the alpha helix structure when it reached this end sequence of amino acids. So this is the model, and there is a software on how to view on the conformation of the amino acid sequence in this MHC protein. This is just an example of proteins that is used for one of the metabolic reactions in biological systems. So note again that the alpha helix can gradually curve or not exactly straight, and that in the beta sheets, the strands are not the same length. You see here, the strands are not at the same length, nor each is perfectly flat. like this. Note also that there are some parts that do not have either alpha helix or beta sheets getting apart. Now those are coils, tawag na to coils. These regions may be disordered or may represent a type of turn structure. Usually they term this one as coil. No? Okay, dili siya. It's not an alpha helix or neither an alpha helix or a beta sheets. So there is what we call as a beta turn. No, the beta turn allows the peptide chain to reverse direction, or allows, or also called as a beta bend, tight turn, or reverse turn. So what is that? So you have 1, 2, 3, 4, getting some turns. You have your amino acid residue here. When it turns, you have your C double bond O and CN. Okay, and then you have your R, CN, C. This is your R, this is your amino acid, but this is your peptide bond. Here you have 1, 2, 3, every 4 amino acids away, no? Then there is what you call as a beta turn. In the beta turn, okay, it is stabilized again by a hydrogen bond. Na beta turn ka no, palayo ka gamay, no? Turn from here. This, okay. And then we also have the 1, 2, 3, 4 amino acid away. Any slight changes here, somewhere here. But here you have, for example, a glycine. The R here is a glycine in a type 2 turn of the beta sheets. So another kind of structure, or in the hierarchy of the proteins, are the ternary and the quaternary structure. So unsa man ang tertiary and quaternary? Okay, it's wrapped up, no, from a secondary structure. Okay, so here is an example of what a tertiary structure is. Before I will define the tertiary, you have the overall structure of your protein itself. Okay, so unsa yang itsura, whether it's globular or whether it's fibrous and so on. So let's define what's a tertiary. The tertiary structure, so it's already mentioned, is the overall 3D dimensional arrangement of all atoms in a protein. No, the 3D, so we'll consider everything, whether na siya, of course, aside from the sequence, mo siya ba siya primary, secondary, or there are other arrangement or conformation of atoms in the overall structure of the protein. So whereas the term secondary refers to the special arrangement of amino acid residues that are adjacent in the primary structures, tertiary structures include longer range aspects of amino acid sequence. So of course, tertiary, because it's tertiary structure na, because long range siya. In the secondary structure, close range pa siya, katong in close proximity of amino acids. Is there a possibility of whether it forms a hydrogen bond formation that would lead to an alpha helix structure, things like that. That will be for the secondary. For the tertiary structure, long range siya. Kita na to, few distance, like how many, like longer than 4 amino acids away, no? What is the conformation of the amino acid sequence? So those is for the quaternary. And then we'll also be considering the tertiary structure, the interaction segments of the polypeptide chains, no? And when that are held in their characteristic tertiary position by different kinds of weak bonding interactions. So we'll consider not just lang katong hydrogen bond, again, that would lead to the alpha helix and the beta sheets. So we'll be considering other kind of weak interactions. Katong akong na-mention kanina, what are those one? Would there be a disulfide bonds? No, disulfide is katong again, the cysteine amino acid and another cysteine amino acid forming a disulfide bond to form a cysteine, katong TIN cysteine. Peptide link, would there be other kinds of interactions. For example, dipole-dipole interaction, katong mga van der Waals interactions. No? So those are what we consider when we're looking at the tertiary structure. Some proteins contain 2 or more separate polypeptide chains or subunits. What we have seen earlier, katong kanino, it's just a one sequence of amino acid. So it can still be entered sa data sheets. No? One sequence from the N-terminus end na start here, and can you find the C on the last of the arrow? Ito na to ang C, N-terminus to the C terminus end. So nag-start data dili, darang C terminus. Ribbon ang end. So nag-start ta here. Okay, so this is the end. So one sequence lang siya of amino acids, but it leads to 2 different conformations and other structures like coil that lead to these structures. But sometimes there can be 2 or more separate polypeptide chains or subunits that may be identical or not identical. And that would lead to what we call as a quaternary structure. Okay, so let's have an example of a kind of protein that it's fibrous. So for example, we'll be talking of a keratin that's we can find in hair, feathers, and nails. The main features of the keratin is that it's very tough, insoluble, protective structures would have varying hardness and flexibility. Of course, it depends on the keratin, where you can find the keratin in your nails, the keratin in some horns or feathers. No, it will have the hardness will vary. And what made up your keratin are mostly alpha helix structures, and from time to time there is this disulfide bonds. For your fibroin protein, it's very soft. Usually you have a beta conformation. And then collagens would have a very high tensile strength and usually have a triple helix. You have a helix triple. So here is the fibrous protein, adapted for its structural function, the alpha keratin. So you can imagine your hair, having a keratin in it. So it assumes an alpha helix structure. And I have mentioned earlier that whenever you have magpakulot, it's actually just a formation of a disulfide bond from a cysteine protein to form a disulfide cysteine, and it will stabilize the firming of your hair. So here is keratin. You have an alpha helix and you have a 2-coiled chain. You have another helix, and then you form a profilament from this coiled chain. Duha ka alpha helix nagsapaw. And from that, you have your profilament like 20 to 30 angstroms away, or it's like 2 to 3 nanometers away. And from that, you actually get your protofibril that would make up your keratin. Okay, so this is your alpha keratin, the structure of your hair. For instance, you have your cells and terminated filaments, and this is where you can find here your protofibril, your protofilament, your protofibril, which is made up of 2 chain coiled alpha keratin. So you have here your alpha helix here. You have your 2 chain coiled coil. You have your alpha helix nga marag natapad, alpha helix you have here. You have your alpha helix, and then duha ka alpha helix that will make your coiled coil trail. From that you have your protofilament. From that you have your a greater number of protofilament, 2 or more protofilaments, 1, 2, 3, more than 2, in a sequence. You have that. Then you have your protofibril, and you have your hair. That's why, if you remember the story, you have it's a very strong one actually, pag baga ang imong hair taas, In fact, nakakatkat sa to katong story nga taas kay yung hair. You have that. So you have more siya og PC kung daghanan nimo ang imuhang sections of your one structure, cross-sectional structure of your one strand of hair here. So that's where you can find your alpha keratin structure. And I have already mentioned that when you have your alpha keratin, the basis of hair sperming of your hair, magpakulot, no? You have your cross-links, the covalent bonds between residue side chains of different polypeptides. If you have your chains of polypeptides here and silingan siya with cysteine residues, and it actually forms a disulfide, and we can actually reduce that one to form your SH free sulfide bonds, or we can curl this one when you have a free one, and we can oxidize again. So it depends on the oxidation reduction process and whether makulot or mas straighten mong hair. Okay. Another structural protein, we have the collagen found in connective tissues, tendons, cartilage. You again see an alpha helix. Yeah, but alpha helix, alpha chain, but you have a chain, alpha chain, but not an alpha helix. This one is left-handed, 3 residues per turn here. And repeating tripeptide sequence is the common one, glycine, X, and hydroproline here. 3 helical strands wrap around each other to form a super helix here. Okay, so this is just a 3D model. I am looking if, hopefully, if it's possible for us to see a 3-dimensional structure of our protein. If it's possible for you, you can download the software where you can see the 3D models of the different proteins in your laboratory. So I'm still looking for that. For your laboratory, this Thursday, I think I'm not sure for the other section. So your first activity will be on the denaturing your proteins, no? From its 3D denaturing. It's the amino acid, and then we'll have an activity on denaturing proteins. So what you'll do is you simply have to watch YouTube for that, or ended message again. Yeah, rocket solve. That's my forget. So anyway, so for your activity, I was thinking if you can do denaturing proteins at home with just some mechanical agents that you can find. Well, your first activity is about determining how to test your amino acids. No? So I'm gonna give the procedures for that, and you will observe a video sa YouTube, and then you'll make your observations sa katong kaan. And maybe in the denaturing proteins, you can perform at home with some instructions that I will give you, and you will do observations, and then after that, you answer post activity session. So again, so this is for your collagen. Again, you can see that like an alpha helix, but not an helix. It's an alpha chain because there's 3 residues per turn lang, not 4 residues away. I don't know, siya ka-share. And here we have another example. You have the self-fibrine. Where can you see that one? You can see that one sa mga spider webs, no? Okay, so you have your spider webs, kang Spider-Man nga web. Okay. Or sa mga spiders, just look at the spider webs at home. If you have spiders at home, and the spider webs are actually made up of fibrin protein produced by, of course, spiders and insects. You have a spider dragline silk, and even now katong mga silk cloth, no, it's prepared by, those are silk, no? So it's made up of fibrins, a protein, katong spider webs. You can find that one. Its structure is largely beta sheets. Now you can see in the structure, you take a small fraction of the strand. Karon pa makakita, participants cannot see. Anyway, back to the discussion, you have your spider webs, and look at the radial strand, one of the strands, and what's in there is actually mostly beta sheets now, but still you can see some parts that's an alpha helix. The rest are like coils, but mostly what you can see are beta sheets. The beta sheets impact strength, and it impacts strength, and the alpha helix imparts flexibility to the strand. So from the secondary, tertiary structure, from the secondary, the proteins or its sequence of amino acids can assume a tertiary structure. From the tertiary structure actually of the proteins, whether it will become fibrous, globular, or membrane, from its overall structure will determine the functions, the particular functions of our protein. So depending kung unsa iyahang overall structure of the protein, its tertiary structure, 3D structure will determine largely what will be the function of a particular protein. So for example, if we see a fibrous proteins, fibrous protein, katong earlier what we have seen, it's a simple regular linear structure. It usually has a role to serve on structural roles. For example, keratin, right? So those are fibrous proteins. So, and usually have low solubility in water or dilute salt solution. So God is good, when we have our hair, kung maligo ta, dili lang kay madissolve ang atong, let's say, hair. So we have our nails. Those are made up of fibrous proteins, keratin, for example. And then if we have our globular proteins, for example, mga enzymes, or what else in the body? We have your hemoglobin, myoglobin. It's compactly folded and approximately spherical in shape. Its function is functions like carriers, wala gibutang diri, but usual functions of globular proteins are carrier proteins, or it can act as an enzyme, catalyze reactions. Another one is you have the membrane proteins, can find in membranes. Usually they have polyhelical structures, and also they are insoluble in water, and usually they require detergents rather, for it to become soluble, or it disrupt the protein structure, require detergents to disrupt this membrane protein. So these are some of the examples. So you have your globular, it's like loosely. You also have your alpha helix, but when you look at the structure, it's globular. You have your fibrous protein here, and here is you have your membrane protein. This is where you can find sa mga membranes, not like on the cell wall, on the cell membrane rather. You have here, those are membrane proteins. Proteins also can be classified based on its secondary structure. Secondary structure proteins. Normally, usually, what are the main secondary structure that would contain our proteins? They can also be defined from its secondary structure, but will lead to the tertiary. So, for example, you have your myoglobin, hemoglobin. Those are globular proteins in its 3D structure. Mostly are made up of alpha helix. You have your immunoglobulin. You will see more of this when you go to your biochemistry, higher biology class, and also your, I think napap mo ikuan sa biology, higher biology. You have here, and you will meet some of these kinds of proteins. Then you have so those are immunoglobulins, maybe made up of beta sheets. Your insulin, it's a hormone regulator. Usually you have your alpha helix, and then your katong mga enzyme proteins like kinase, phosphorylase, toxotin. We have your alpha helix and beta sheets in there mostly. So here it's not important, no, they can alpha protein, beta protein. Alpha protein, pag they're usually made up of alpha helix, beta protein, they usually made up of beta sheets, alpha beta protein, if they have alpha and beta, or alpha slash beta, if you have an alternating alpha and beta sheets in the segments, like the one that you have seen earlier. Okay. So coil protein, if you have no regular secondary structures, no alpha or beta, katurang coils, you see in the initial structure. So what you see in the tertiary structure, so again, this secondary folding here, secondary and tertiary closely related to each other, no, but in the tertiary, we are looking at the long range, as I mentioned earlier. So we're looking at overall 3D structure, not just the alpha or the beta, but the overall. In the secondary, my good, we're just focusing on the particular short sequence of amino acids. The tertiary, we'll be looking at the long range, and this is the 3D model of what a tertiary structure is. This is a space filling model. This is a ribbon model, surface contour model, and mesh model. So they can be seen via software. I will be looking again for a particular one that will be much user-friendly for you for beginners. Okay, so here you have a ribbon structure, which is more in the ribbon structure where you can see the alpha helix structure here. Okay, so this is a 3D model. So 3D model, you can actually see in the tertiary structure, you can still see your secondary structure, but you couldn't see what are the particular amino acid sequence unless we see on katong, we really identify what are the amino acid sequence here. But again, we have to remember that when we're looking at a particular function of the protein, we're simply, the overall function of the protein is determined by its tertiary structure in its folded state rather. So here there is a particular binding site here. What is this? I think this is part of a hemoglobin, or you can see a hemi group here, and these are some other group of atoms here that is not included in the tertiary, in the secondary structure here. These are the hydrophobic side chains here, the blue one are the hydrophobic side chains. So here we have other globular proteins. You have the cytochrome C, lysozyme, ribonuclease. These are also enzymes. The common features of globular proteins. And this is the mesh structure. When you see, like, it's more like a space filling model of how it would look like, no? It's like a globular, and their functions are enzymatic. So what stabilizes the tertiary structure? I will end, and can we call again? Can you call again? I will end this call, and let's meet again for So what stabilizes the tertiary structure? So one, we have the covalent bonds. The covalent bonds most often involved in stabilization of the tertiary structure.